right, hello everybody. I hope you're feeling a little bit refreshed after this short break. My name is Mareike Grotheer and I'm a junior group leader at the Philipps University in Marburg. I would like to talk to you now about a very special use case of tractometry, which is to apply it to human infant data. Right. And for my talk, here's a brief overview. I will start with a short motivation as to why it might be relevant or interesting to apply tractometry to infants. Then I will briefly explain the term tractometry. This you've already heard, but it's always good to have a short repetition. And then I will slowly build up a tractometry pipeline that you could use to analyze infant data. For this, I will start with an adult pipeline, but then tweak it so that it will work for infants. And each of my slides will have little blue boxes, such as the one that you can see here on the right. They're not immediately relevant to the content of my talk, but rather points out resources for further reading and other helpful links and tips. During the talk, I would also like to pose a few quiz questions, and I would be really excited if you would participate in the quiz. So in order to participate in the quiz, simply pull up the link that is provided here, and I'm actually going to pause here for a few seconds to give you a chance to navigate to that link. And if you haven't quite made it there, don't worry, the link will come up again later in the talk as well. So first of all, why might be worthwhile to think about doing diffusion-weighted MRI in infants? To answer this question, I would like to show you a brief video. In this video, I'm comparing changes in the overall brain volume between the first and the second year of human life. And if you look very closely at this video, you might notice that the brain grows much more rapidly in the first year of life compared to the second year of life. In fact, the brain never grows as rapidly as it does during early infancy. And to me, the fact that the brain changes so rapidly makes infancy a particularly interesting developmental period to study. And the brain doesn't only just grow rapidly during infancy, but these volumetric changes are also associated with important and tightly regulated microstructural changes. So in this graph here, I'm plotting changes in R1 over the course of the human lifespan. R1 is a measure which is related to myelin content. And the blue shading basically indicates approximate growth rate. And as you can see, myelin grows particularly rapidly in the first year of life as well. Myelin is also associated with various different psychological and neurological disorders. So understanding how myelin is usually built up in this critical period where it's growing really rapidly is important for our understanding of human brain health. So based on what I showed you so far, I hope I could convince you that studying infants is particularly exciting. Their brain not only grows rapidly, but there are also several psychological and neurological disorders that already emerge in infancy. And if we can detect these disorders very early on, this may also help us to intervene. More broadly, there's just not that much known about the infant brain, so there's still a lot of exciting open questions for you to tackle. And most importantly, you do not need to be equipped to collect infant data yourself, because there's now more and more large-scale data sets that contain infant data that are freely available that everybody can play with. And I'm listing a few of them here in this blue box. The best well-known example of this is probably the data of the Human Connectome project, which contains thousands of infants. Okay, I see this is working, that's great. Just to drive home how new tractometry in infants really is, here's my first quiz question. What do you think if a PubMed search with diffusion and bundle in adult gives 1,500 results? How many results do you expect for bundle and infant? I see there's a little bit of a debate going on, which is great. Nice, you're converging actually on the right answer. So the correct answer, I did this recently, is 150 results. Which is just really significantly less than what you can find for adult data. So what is tractometry, and why do we want to apply this to infants? This you have heard before, but again, repetition is always good. The term tractometry describes an approach in diffusion MRI where first you would identify the large-scale bundles or pathways of the brain that you can also think of as highways that connect distinct parts of the brain. Once you have here in this example, for instance, I'm showing you the arcuate fasciculus that was identified in the brain of a newborn. Once you have identified the bundle, you can then analyze its microstructural properties. And in tractometry, you would analyze these microstructural properties at various steps along the length of the bundle. So rather than getting a single mean value for the entire bundle, you see how your measure of interest, such as, for instance, FA, changes along the length of the bundle. Because of this, it's also sometimes referred to as a long tract analysis, and the resulting graphic that I'm showing you here is often called a bundle profile. So once you have such a bundle profile, you can start asking interesting questions, such as how does it change when an infant gets older, which is what you can see here. So just to illustrate what one could do when applying tractometry to infants, here are a few examples that I pulled relatively randomly from the literature. For instance, Wilson and colleagues identified white matter bundles in utero, so before the infants were even born, and then evaluate their microstructural development in utero as well. Dubner and colleagues, on the other hand, looked at white matter properties at birth and found that these properties actually predict the 2-year language outcomes of the infants that were born preterm. And then in our most recent study, we found that in humans, myelin actually grows more rapidly in utero compared to ex utero, and that this slowdown in myelin development at birth explains why infants that are born preterm show a delay in myelin development. So that was by no means an all-encompassing list of what is out there in the field. There's lots of cool research for you to discover, but I hope that these examples could tweak your interest. So hopefully now you're interested in doing tractometry in infants. How would you go about actually doing that? So for the rest of my talk, I will build up a tractometry pipeline that will work for infant data. And again, I will start with an adult pipeline and really highlight some of the issues that you might run into when trying to apply this pipeline to infant data. So here you have your typical adult diffusion analysis pipeline. There are various different tools and approaches that you could do to get it to probably the similar solution. This is one pretty standard pipeline. So first, and we will go over each of these steps in detail, but it will probably include some kind of pre-processing. Then you will generate fiber orientation density functions from which you can then generate a kinectome, a tractogram, which you will, by using some kind of tool, dissect into bundles, and then you can generate a bundle profile. The first step in this pipeline is to pre-process your diffusion data. As a minimum, this will usually include correcting for susceptibility-induced motion, distortions, motion correction, and eddy current correction. And out of all of these steps, motion correction is one that is particularly challenging when it comes to infant data. Because, of course, we can't ask the infant to please lie still in the scanner. And even if we could, the same amount of motion will be more detrimental to data quality in infants because their brains are just so much smaller. So when it comes to motion, I think one of the best ways to go about it is to prevent it from the get-go, so at the time of data collection. And even though we can't ask the infant to please lie still in the scanner, there are still many little steps that we can do to substantially decrease the infant's motion. First, you would typically want the infant to be naturally sleeping during the scan. And in order to encourage this, it is helpful to scan at night during the infant's natural bedtime. It's also helpful to feed the infant right before the scan so that they sleep more readily. And sometimes you might also want to swaddle the infants or even use a professional immobilizer. There are now also infant-specific MRI head coils on the market that tightly fit the infant's head and actually have a good impact on data quality. These head coils also have space for headphones and other ear protection, which of course is not only important for the infant's sleep quality, but also for their overall well-being. But most of all, what is important, I think, is that you monitor your infant very carefully. And as soon as your infant shows signs of waking up, it might be best to stop and start the scan over at a later point. So in order to collect high-quality infant data, you will need a lot of patience. But all that said, as with all MRI scans, there will always be some motion left over. So when working with infant data, you just want to make sure that you're utilizing the latest approaches in motion correction. For example, you could use the newest version of FSL's EDDI tool, which performs motion correction in 2 steps. First, it does outlier replacement, which means that it replaces every slice whose average intensity is lower than a certain standard deviation from the expected intensity. And this is because motion will lead to signal dropout in the diffusion image. And in order to sort of get rid of that, EDDI will remove slices that have very low signal. In the next step, it will then check if slices within one volume are aligned to each other and try to realign them using a processor similar to rigid body alignment. I would also recommend paying attention to the output of your motion correction, as this will help you decide if some data may need to be excluded due to excessive head motion. For example, EDDI will tell you what percentage of slices were replaced and how much motion there was in each of the different directions. So this will allow you to make informed decisions whether some of your data may need to be excluded or not. So once we have preprocessed the diffusion data, next we want to generate a connectome or tractogram. For this, we will use an approach referred to as multi-shell, multi-tissue constrained spherical deconvolution. There's obviously a lot to unpack there, so let's start with constrained spherical deconvolution. Each white matter tractogram generally consists of millions of streamlines that pass through the brain and hopefully represent a model of what the actual fibers of the brain may look like. In order to draw these streamlines, we need to understand what the diffusion looks like in each voxel, so that we know how streamlines may travel through the voxel. In the classical approach, which is called diffusion tensor imaging, the diffusion within a voxel would be described by a single tensor. The tensor tells us what is the mean direction and the mean intensity of the diffusion in each voxel. However, and this came up earlier in the talk, most voxels in the brain will actually have multiple fibers that will go in different directions. And tensor model does not capture such crossing fiber regions well because it only gives you one single value for each voxel. So constrained spherical deconvolution is a slightly more sophisticated approach where we decompose the diffusion signal into a number of individual fiber orientations or FODs. Thereby, we are better able to deal with crossing fiber regions and to trace more accurate streamlines through the brain. Importantly, in order to better match myology, we want our streamlines to be limited to the white matter. Hence, in multi-shell, multi-tissue constrained spherical deconvolution, we will generate FODs separately for the white matter, the gray matter, and the CSF. This, however, leads me to another infant-specific problem, which is that in infants, there's very little myelin in the white matter. The myelination has really only started in a newborn. And this makes it difficult to distinguish between different tissue types of the brain. Okay. So the question that I would like you to answer is, in a T1 weighted image of a newborn, what is brightest? The white matter, the gray matter, or gray and white matter are equally bright? I like that there's a bit of a debate going on. In a newborn specifically, the correct answer is actually the gray matter. And this is what you can see here. So in a T1 weighted image of a 2-week-old, the white matter is actually darker than the gray matter. This is because the white matter contains very little myelin. And as the myelin content increases during the first year of life, the contrast between the gray and the white matter slowly shifts, so that by 12 months, the white matter is actually lighter than the gray matter, which is what we would usually see in adults. This, however, is an issue for us because in order to fit separate FODs for the gray and the white matter, we need to be able to draw boundaries between these different tissue types. In adult diffusion-weighted imaging, we often use a cutoff in FA to determine what is white matter and what is gray matter. as in adults, the highly myelinated white matter has a higher FA than the gray matter. However, again, since infants' white matter contains very little myelin, there's also no big difference in FA between the white and the gray matter. So also this approach will not work very well. And just to demonstrate that, here's an example. So in this image that you're seeing here, the white matter, gray matter, and CSF were automatically cemented based on differences in FA. And as you can see in this adult data set, this is working really well. So the blue areas are the white matter, and that pretty much matches what we would expect to see. If we use the same approach in infants, it sort of looks like this. So you can tell that this probably did not work very well, because now it considers only these really small blue areas as white matter. And if we were to use this to perform tractography, we would only find streamlines in this blue part of the brain, which is not what we want. We could sort of tweak this a little bit by changing the FA threshold as what would be included as white matter. But this will never get us all the way there, because the FA between the areas where the FA between the gray and the white matter are just simply indistinguishable. And in addition to that, the brain is developing so rapidly in this age range that we would need to find a new FA threshold for each age group, which is not very practical. So instead, what I would suggest is to not even, at this stage at least, not even try to separate out the white and the gray matter, but treat everything that is not CSF as white matter, which now in this picture is green. What we end up with if we do that are nice FODs for each white matter voxel in the brain. So this will allow us to trace accurate streamlines through the entire white matter. We're not quite there yet though, because now we have not separated out the gray matter. So our streamlines will go all the way into the gray matter, and as we have already heard before, tractography does not yet work very well in the gray matter, so we don't really want to do that. So to overcome this, we can leverage an approach that is referred to as anatomically constrained tractography. For anatomically constrained tractography, you would also need to collect an anatomical image of your participant. For infants, this will usually be a T2-weighted image. You can then use automatic approaches like iBeat or infantry surfer to segment this anatomical image into white and gray matter. Assuming that your diffusion data and your anatomical image are well aligned to each other, you can then use this boundary between the gray and the white matter that you got from the anatomical image to constrain your tractography. So in this way, you will end up with a tractogram that has streamlines that end at the boundary between the gray and the white matter. And once we have such a nice connectome or tractogram, the next step for us will be to identify the large-scale bundles of the brain. Here, however, we run into our last infant-specific challenge, which is quite an obvious one, that infants have much smaller brains than adults do. And this does cause problems for us, though. So just a reminder, in order to perform tractometry on any kind of data, first we need to identify the white matter bundles of the brain. And we can do so manually by basically chopping away at the connectome until we have the bundle that we're interested in. But this is very labor intensive and it takes a long time. So for me personally, it takes at least an hour per bundle and per subject. So I wouldn't want to do this for a lot of subjects or very large data sets. In addition, by defining bundles manually, you're also somewhat reducing the reproducibility of your results. So it's generally advisable to use automatic approaches to do this. And the good news is there's various different, very cool automatic tools for you to choose from that will segment the connectome for you. The one thing that you should be wary of when you're trying to do this on infant data, though, is that these tools were designed for adult data, and they don't necessarily immediately translate to the infant brain. So the automatic tool that I am going to talk about today is AFQ, or Automated Fiber Quantification, which you have already been introduced to today. So AFQ has been developed by Jason Yeatman, and it's now available both for MATLAB and Python, which is very cool. And again, this picture you've already seen, but just to recap, AFQ identifies white matter bundles by leveraging two waypoint RIs for each bundle. A streamline becomes a candidate for being part of the bundle if it passes through both waypoints. There can, however, be situations where a streamline passes through the waypoint RIs for two different bundles, and AFQ solves such issues by also using a probability map of each bundle. In this case, a streamline will be assigned to the bundle that it overlaps with most strongly, so that in the end, each streamline is only assigned to a single bundle. After that, with the resulting bundle is often cleaned, it's sometimes clipped to in between the waypoint RIs, and then the microstructure properties of interest are measured along or analyzed along the length of the bundle. Most of this will work just the same in infants, however, there's one important problem, and that is that the waypoint RIs and the fiber probability maps are defined in a commonly used adult template brain, the MNI brain. In a typical adult pipeline, the individual subject data is aligned to the MNI brain, and the inverse of this alignment is used to bring the waypoint RIs and the probability map back into individual subject space. However, as the infant brain and the adult brain are quite different, particularly in terms of brain size, this approach of aligning to the MNI brain might not work very well. So what do you think? The total brain volume of infants younger than 1 month is 17% of adult volume, 36% of adult volume, 55%, or 82% of adult volume. Nice, the majority of you is correct. It's 36%. And just to illustrate this a little bit more, here's the B0 image of one of our newborn participants. If we were to use AFQ as is, we would align this B0 image to the adult MNI brain, which looks like this. Given that both of these images are at the same scale, this looks like a very difficult alignment to make. And maybe this is not a very good way to go. A much easier alignment can be achieved by aligning to an infant-specific template brain. So for instance, you could use the University of North Carolina neonate template, which I believe has been made from over 100 typically born newborns. And as you can see, this will require a lot less stretching of the individual brain, so it's probably a much easier alignment to make. In my work, an additional step that we usually do is that we align the individual infant's diffusion data first to their T2-weighted anatomical data, and then we align the T2-weighted data to the template. And the reason why we do that is that the T2-weighted data just generally has a better contrast, so it's a little bit easier to align. But this step is probably optional. So now you might wonder if this difference in alignment actually has an impact on the bundles that you can identify. And one way to basically answer this question is to first start by defining your bundle of interest manually. This is what you can see here. For one example bundle, this is the forceps major, a bundle that connects the two hemispheres of the brain. And as I mentioned before, these manual bundle definitions are very labor intensive, but they do not require any form of alignment, so you can consider them as a gold standard for what we're trying to do here. Now, once we have these manual bundles, we can compare our automatically identified bundles to these manual gold standard bundles. And what you can see is when you do this and you do a different type of alignment, it does actually impact the type of bundle that you can identify. So in this particular example subject, you actually cannot identify the bundle at all when using the MNI brain. But when you align the data to an infant-specific template, you can recover the bundle quite well. And in this case, it seems pretty reasonable to conclude that using an infant-specific template is actually good for your data quality, but we also wanted to quantify this somehow across multiple subjects. So for this, we performed a dice coefficient analysis, which is basically a measure of overlap between the manual bundle and each of the automatic approaches. A dice coefficient of 0 would mean that there's no overlap between the automatic and the manual bundles, and a dice coefficient of 1 would indicate that there's perfect overlap. And as you can see, using an infant-specific template, which is what is called baby AFQ here, will improve the overall quality of the bundles that you can identify. And baby AFQ is actually a software package that is available both for the MATLAB and the Python implementation of AFQ. So you can just use it for free, and I hope that this will be helpful for you. And baby AFQ also includes a few additional changes, such as changes to the waypoint RIs. Again, these changes were made to handle the much smaller brain size of the infant brain. For instance, many of the waypoint RIs are just more spatially restricted in baby AFQ as opposed to regular AFQ, as we found that this helps with the identified bundles. As you can see in this graph, overall baby AFQ performs better than regular AFQ for almost all the bundles. And this is true regardless of whether you use the MATLAB or the Python implementation of the tool. And most importantly, I hope that this illustrates the importance of using infant-specific tools for infant bundle identification. And luckily, more and more tools are becoming available to you. So instead of baby AFQ, you could, for instance, also use Tractometry, which is the infant-specific implementation of Tracula that we have already heard about today. So now that we have nice bundles in each of our infants, we can start analyzing and comparing various microstructural measures along the length of the bundle. Importantly, this last step does not require any infant-specific changes anymore because now we have resampled the data to a certain number of steps along the length of the bundle anyways. So we have sort of moved out of the infant-specific brain space. So just to recap, this is my last quiz question for today. Which tractometry steps are particularly challenging for infant data? Motion correction, tractography, and bundle profile generation, eddy current correction, tractography, and bundle profile generation, motion correction, tractography, and bundle identification, or eddy current correction, tractography, and bundle identification. It's actually the third one, so I guess this one is a little bit misleading. So what I meant with bundle profile generation is really just this, like, creating this graph where you have resampled the data to a certain number of nodes. So there we have moved out of the infant-specific brain space, so it's not so hard anymore. But the bundle identification is what's really tricky because the infant brain is so small. Typical adult pipelines will not work well for us in that case. So in conclusion, infant tractometry is a comparatively novel field with much to discover, whereas it will be most successful if pipelines are specifically adjusted to the infant brain. And specific challenges that you should maybe be aware of when doing tractometry in infants include motion, low myelin content in the white matter, and an overall smaller brain size. Thank you.
Thank you. Okay, first question here. The gray matter, white matter segmentation did not seem to identify the deep gray matter. Is that a problem for the automated tractography?
You can feed in labels for the deep gray matter as well. For my purposes, I didn't do it and it worked out fine. But if that's something that you're interested in, specifically also maybe connecting two areas in the deep gray matter, the tools allow you to look at that as well.
What should be the ideal resolution for the scans for good tractography?
I'm going to go with Arial for this. More data is always better. I mean, I will say that the infant brain is smaller, so this is something to keep in mind when you think about your resolution. So you might want to opt for a greater resolution than what you would usually do with adults, but also your scan time is very limited with infants because as soon as they wake up, your scan is over. So it's a hard balance to strike. So I'm not going to give you a number here. Just get as much data as you can, I suppose.
I'm going to ask a question that I couldn't type in while we were talking, but it seemed like the regular AFQ actually performed reasonably well for some of the tracts and then quite poorly for others. Do you have any insight into why some areas do well and others don't?
Yeah, that's actually definitely something we have seen. I think one intuition that I got is that particularly curvy tracks are really hard. So the arcuate fasciculus took us a really long time to tweak until we got it properly, which might also be because there's so low LFA that it's just really hard to trace these kinds of curves in general, and then you just don't get good bundles that way.
Okay, thank you. I don't think there's any more questions right now, so thanks very much.
Thanks, everyone.